The third macromolecule are proteins. And proteins are a huge amount of our body, and they have a huge variety of functions. So more than 50% of the dry mass of most cells. That's pretty huge if you think about it. More than 50% of all of your cells means that more than 50% of your body is made up of proteins. One specific type of protein that we're going to talk a lot about this semester are enzymes. These are catalysts. So a catalyst is something that is going to take a chemical reaction that would happen normally and speed it up so that it's a metabolically effective reaction. Proteins do have specific monomers and polymers. So the monomer is called an amino acid, and there are 20 different versions of this. They all have the same basic structure, and when we put them together in a chain, we use our dehydration reaction to build them together. We come up with a polypeptide. So poly meaning many, peptide is generally going to mean protein. So this is a polymer built from many amino acids all put together in the correct sequence. A single polypeptide might be enough to make a protein. Sometimes a protein actually needs multiple polypeptides stuck together to create the right shape for function. So the protein is the final functional molecule, and it either has one polypeptide or more than one polypeptide, which means a functional protein can either be one polymer or two polymers or three polymers or four polymers together working as one. So let's go through some of the different functions. I already talked about enzymes. So an enzyme is going to speed up a reaction. So in this case, it is going to break a bond. What it does is it brings that into the protein here, and it helps to facilitate that reaction by breaking this bond and making that go faster. We have defense. So your body has protection against disease in the form of antibodies, which are made up of proteins. And these antibodies are little Y-shaped molecules, and they recognize pathogens like bacteria and viruses, and then they are able to attack those more effectively, collect them up for your body to get rid of. This is what we're trying to stimulate with the COVID vaccine, is to stimulate your body to make the antibodies to the coronavirus. We also use proteins for storage. So one example is milk. We have albumin, which is found in egg whites, and these are storage sources of amino acids for developing infants. Milk in the case for babies, giving protein to babies. Albumin in the case of eggs to develop an embryo that's inside of an egg. We use protein for transport, so this is how we move things around. So here you can see there's a cell membrane. There's some molecule here that is not going to be able to cross that membrane on its own, probably because it's got some kind of charge and it can't get through that hydrophobic region here. And so what it does is actually moves through a transport protein, and that's going to allow it to move one way into the cell there. Some more examples of transport is hemoglobin. Hemoglobin captures oxygen from the lungs and it carries it through your blood. So hemoglobin is a protein that oxygen attaches to, and then it moves through the body, and once you reach a place in your body that has low oxygen, then you're going to release that oxygen from the hemoglobin to give oxygen to all of the body parts. So instead of just transport across a membrane here, this is talking about transport all the way through your circulatory system. We have hormonal proteins, so some hormones you saw were steroids like estrogen and testosterone. Another hormone, for example, insulin and glucagon, that are used to regulate your blood sugar are actually proteins, and so those move around in the body, dissolved in the blood. A receptor, so this is something we'll talk about in chapter 11, is how we receive signals. So you can see that on the outside of this cell membrane here, we have these little receptors. They almost look like TV discs that are receiving a TV signal. And what they're doing is they're receiving these little signals, and that's going to send some information into the cell. Proteins are involved in movement. Protein is typically found in meat, and meat on your body is your muscles. So we have muscle tissue, which are going to be high in protein, as well as your cytoskeleton. Your actin and your myosin molecules inside of cells are contractile motor proteins as well. And then protein is also used for structural support. Keratin is a specific protein, and that is found in your hair and skin and nails. That's what helps to make you waterproof. And then collagen is another protein that is found in the skin. So collagen is going to give your skin a structure to it. When collagen tears, it actually creates what are called stretch marks. And so that's because we've changed the shape of the skin so quickly that it actually tore those proteins. So here we're going to take a closer look at the amino acid monomer. So the monomer is the amino acid, and they have specific parts that all amino acids have. They all have a central alpha carbon here, and around this carbon they have 4 different things. The first one is a hydrogen, so every amino acid has 1 hydrogen here. They all have an amino group, so an NH2 amine group, which if you remember acts like a base. And then they all have a carboxyl group, that COOH group, which if you remember acts like an acid. So proteins actually make a really good buffer because they have a base and an acid. This part of a amino acid is the same for all 20. They all have an amino, a hydrogen, a carbon, and then a carboxylic acid. This part is variable. So the R group is a different group. It's called the reactive group for R. So this is what's going to give each amino acid its different properties. This could be as simple as just a hydrogen, or it could be a large complex molecule. So when you look at this slide, you can see all 20 amino acids, but you do not have to memorize these. So I'm just showing you this slide to illustrate the point. You can see in purple on all of them, they are exactly the same. So the purple part was that amino group, the hydrogen, the carboxylic acid. All of them have amino, hydrogen, carboxylic acid. And then the other colors, the yellow, the green, the pink, and the blue, are going to be different R groups. You can have R groups that are nonpolar, so all of these are generally going to be nonpolar proteins. So if you put a lot of these together, they would make a nonpolar region that could fit really nice in a cell membrane. These side chains tend to be polar, so if you look at these have nitrogens and carbons in them, as well as oxygens, which is going to cause polar covalent bonds and polar molecules. These are going to dissolve well in water. These are going to orient towards the water. We've got ones that are acidic, so you can see some carboxyl groups here that are going to give these an acidic nature. And then these are basic. You can see some amino groups here, which are going to give those side chains some basic properties. And so the functional properties of a protein depends on which amino acids and in what order it makes it up. So again, please don't memorize all 20 of these. What I want you to recognize is that this part is the same, and that this functional group here, this R group, is going to give it its functional difference. So a polymer is the grouping of amino acids into a chain. The dimer we call a dipeptide, so it would have one amino acid bonded to another amino acid, and that bond right here is called a peptide bond. So the covalent bond for proteins is called a peptide bond. Thus we have a polypeptide, which is going to have many peptide bonds and is going to create long polymers of amino acids. Polypeptides can vary greatly, which is kind of the theme of this entire group. So they have a vast diversity of functions, so they must have a vast diversity of structures. Within a single protein, there are 4 levels of structure. So the primary level of structure is just the order of the amino acids. This amino acid, this one, this one, this one. So we give these letters. These are basically like words in a sentence, and these are held together by that peptide bond. The secondary structure is, now it's not just the order of these amino acids, but we're going to start to see some 3D structures. They're going to form themselves into sheets or coils, depending on how the primary structure organizes itself. These orientations are typically going to be formed from hydrogen bonds. The tertiary structure is the interaction among side chains. So this is going to be a 3D folding. And this is going to be caused by those sulfur cross-linking. And then the quaternary structure, not every single protein has a quaternary structure, but this is when we take more than one So not every protein has a quaternary structure. This is going to happen when we have multiple polypeptide chains. So if I have one protein here, or one polypeptide, and then there's another polypeptide chain here, and this is my final protein, then the quaternary structure is that we have 2 polypeptide chains together. So what I'm going to do is show you each one of these in a little bit more detail. So primary structure is the sequence. This is like the letters of a word. So you can see that each amino acid has a name: glycine, proline, threonine, glycine, glucine, serine, and so on. And so each single one of these is put in this specific order. If we took the letters of a word and we mixed them up, it doesn't mean the same thing, right? These two words have the same letters, but they're in a different order, so they don't mean the same thing. So if I take these amino acids and I decide to switch them around a little bit, the primary structure is going to change. So this primary structure is determined by the genetic information. Our DNA forms a molecule called RNA, and then the RNA template tells us what order to put our protein amino acids in. AA for amino acid. Here's the secondary structure, and you can see the 2 types of secondary structure that we have here. This is the alpha helix or the beta pleated sheet. So when we zoom out on a structure, you can see there are regions that are helical, and you can see how this is maintained by those hydrogen bonds between the different regions of the protein. There's also regions that look a little flatter, and those flat regions are these pleated sheets, and those pleated sheets again are maintained by the hydrogen bond. The tertiary structure is the overall 3D shape. So this is the 3D conformation or structure of a whole polypeptide chain. So this is going to result in interactions between the R groups. So again, we're going to see some of those sulfur linkages and other interactions that are going to help hold this together in its correct 3D structure. If you change this 3D structure, this protein is not going to function the way it is supposed to. And then our last level is the quaternary structure, and this is when you have more than one polypeptide. So this example is hemoglobin, and it has 4 polypeptides, an alpha subunit and beta subunits. So you can see in blue, you've got 1, 2 alpha subunits, and then in teal here, we've got 1, 2 beta subunits. So this entire protein is made up of 4 polypeptide polymers that are stuck together into this hemoglobin shape. Now that we've reached our quaternary structure, this is going to be a functional molecule, and it can bind some iron, and the iron can bind on oxygen and carry oxygen throughout your blood. The primary structure is directly related to the secondary, tertiary, and quaternary structures. A slight change in one single molecule. So number 6 in the normal hemoglobin is glutamate, and number 6 in this mutated version is valine. And that's literally can be a single mutation in a DNA that changes just one letter in this big long word. And what happens is the beta subunit ends up having a different tertiary and secondary shape. And then our entire quaternary structure ends up having a different shape. And then because it has a different shape, it has a different function. So an overall red blood cell with normal hemoglobin has this kind of discoid shape with no nucleus in the middle, and it's round and it's going to flow through your body really easily. But when we have this mutation, we end up with sickled red blood cells. And you can see here that those are able to bind with each other really easily to form these large clots and fibers. They're also not as good at carrying oxygen, and they can cause a lot of pain and bruising and damage. One mutation in a single part of the DNA changes one aspect of the primary structure can have a huge impact on the overall function of the quaternary structure of a protein. We can't really predict the protein structure, so that'd be tertiary or quaternary, from its primary structure. And that's just because there's so many interactions in the environment in your cell that cause it to form the correct 3D shape. If we change any of the environment, so if we have a really low pH, or really high salt concentration, or really high temperature, something like higher than boiling, those environmental factors can alter the way that the protein is folded and cause it to denature. So a denatured protein looks like this. It is unfolded. This is what it should be normally. And when you, let's say we put this in like a pH of 1, when it's supposed to be at a pH of 7, what's going to happen is it's going to denature. Or this might happen if we put this at a really high temperature, like 120 degrees Fahrenheit, when it's supposed to be at, 98.6. And what happens, and it becomes biologically inactive, and it typically cannot be refolded. So once a protein is denatured, it's not going to be able to be turned back in. This is actually what you do when you cook your food, is you denature all of the proteins inside of your food. So if there was anything like bacteria, salmonella, stuff like that, that you denature it and it can't harm you. We do have human diseases such as Alzheimer's and Parkinson's that are associated with misfolded proteins. So there's beta amyloid plaques that build up in Alzheimer's that cause clogs in the brain. Mad cow disease is associated with misfolded proteins called prions. So denatured or misfolding of proteins can have a pretty drastic effect on the body. So here's that overall structure. Make sure that you know the amino acid monomer and the structure of those. Again, don't memorize all 20. Make sure that you know all of the different functions and examples of types of proteins, as well as the different structures: primary, secondary, tertiary, quaternary. And then the covalent bond was the peptide bond.